We're very happy today to receive Ralph Scheinfeld as a speaker. Ralph is a very good friend for a long time, since I don't know, 15 years, 2005, the same date, like in UCLA. And I met Ralph in a conference, and a few years after we worked together, I was his postdoc in Uppsala University. He is here in the Cal Street corner to have this exchange professor to the universities. Is very nice. We have this opportunity. If someone has something to talk with Ralph, just let me know. He stay here until 10 next week. Ralph, welcome to welcome to.
Thank you, Ruby.
Agora sim.
Oi?
Sorry.
Is it okay?
It's okay.
Okay. Okay, so as I was saying, so the topic of today's talk is nano-devices for DNA sequencing. But before I get to that, maybe just a very brief introduction about the place that Rodrigo mentioned, Uppsala University. So Uppsala, the city, is in Sweden. It's very close to the capital, Stockholm. And the university there actually is one of the oldest universities in the Nordic countries of Europe. It was established more than 500 years ago. And because it's so old, it doesn't really have a central campus. The departments are spread out all over the city. And we are located there on the, little bit outside the city, in the Ångström laboratory. The Ångström laboratory is named after father and son Ångström, who were two scientists who were working there in spectroscopy. And this is how the building looked like a few years ago. They're always constructing and adding new wings to it, actually. And so there's some latest additions to it. But it houses the different kind of department of chemistry, department of mathematics, information science, and also department of physics. And the department of physics and astronomy, as you can see here, is divided into several divisions where we're covering, in principle, all the areas of physics. I'm working in the division of materials theory, and there you can also sort of subcategorize the research that you're working on into computational physics, where I'm working in mathematical physics, a branch that is just focused on materials for sustainable energy solutions. And then we're also doing some code development, so we're also developing new algorithms to implement new electronic structure methods. Okay, back to the title slide. So I want to talk about this topic here, and DNA sequencing, just to put everybody on the same page, just a very brief primer or very brief introduction to DNA. So as you know, all the life on this earth has something in common, is that the information about how to build this organism and how to run this organism is encoded in this DNA. And for a cellular organism in the cell, we have a nucleus, and then in the nucleus of the cell, tightly wrapped up in these chromosomes, we have these DNA molecules. And the DNA molecule is made up of 4 different bases. A, C, G, and T is the abbreviation. And the sequence of these bases tells, in principle, everything about the organism, how to build it, and then how to operate it, how to run it also. So if you're a life scientist, you want to study living organisms, I mean, the starting point could be that you sequence the genome, and then you get a lot of information out of that. And so life scientists need tools to determine the sequence of the genome, and they had this tool for a long time, since the late 70s of the last century. There's a method called Sanger sequencing, and basically it can tell you, without going into the details here, the order of these base sequences in a DNA strand. The only problem with this method is that it works only relatively efficiently or well on short strands of DNA. If you have very long strands of DNA, you need to break them down into small pieces, and that means for something like the human genome, where you have 3 billion bases in a row, you need to really make it into short, cut it down into short strands, sequence those with the Sanger sequencing method, and then stitch together a puzzle that has basically 1 million pieces. So you can imagine that with this method, it's a very big undertaking, a very huge effort to sequence the human genome. But at the beginning of this millennium, or at the end of the previous millennium, whichever way you want to see it, this was actually accomplished. It was this Human Genome Project, where for the first time, the 3 billion letters of the human genome, approximately, were actually sequenced. So we had some blueprint of what distinguishes humans from other animals, basically. But it was a huge effort. It took years, it took a lot of labs that are working on it, and it took hundreds of millions of dollars, or even billions of dollars, to achieve this. But then, as with any kind of technology, humans are always trying to improve the technology, make it more efficient, make it faster, make it cheaper. And DNA sequencing is no exception. So you see here how the cost of sequencing these 3 billion bases of the human genome has dropped as a function of time. And I want to notice that this is a logarithmic scale. So this has dropped over 5 orders of magnitudes until today's time. And now it is possible to sequence a human genome for something like $1,000 and in less than a day. You can do it with one machine, one person can in principle do it. And this is something that was unimaginable maybe 20 years ago. So some of this progress, this technological progress, was driven by nanotechnology, actually. And so at first, you might be wondering, nanotechnology and life sciences, how does that really go together? And in life sciences, you study living materials, and nano is this kind of inorganic dead materials. But if you just think from a purely length scale point of view, the two actually go very well together. So if you take, for example, DNA, the characteristic length of the features of DNA are of the order of nanometers. So if you want to probe a DNA molecule, a nanomaterial, a nano-device is ideally suited in terms of the length scale. So you have really the resolution to look at individual bases if you have something that is of nanometer size. So how to actually do it then? How to use nano-devices? In particular, I will focus on nanopores to sequence DNA. So I will run through some kind of schematic setup here, some experiment in a cartoon way, just to illustrate the idea. So we start with some empty container, some box here, and we fill it with water. And then we put ions inside. For example, sodium and chloride, but it can be also potassium and chloride, some other ions also. Now, if we stick electrodes inside our container, positive and negative electrodes, and we have a voltage source and an ammeter, and we close the circuit, then what happens is that these ions will be attracted to the electrodes that have the opposing polarity. Okay, and we measure some kind of a ionic current with our ammeter. Okay, that's not so interesting. Let's introduce the nanopore now. So in a top-down view, I take some kind of a insulating membrane, something that doesn't conduct electricity. And I will tell more about how to drill a nanopore inside, but basically for now, all you have to know is that it's some kind of an opening that has a few nanometers in diameter, nano-sized opening. In reality, it would look something like this. So this is a picture taken with a transmission electron microscope. There's a nanopore fabricated in silicon nitride, and the diameter is around 15 nanometers, just to give you an idea what it really looks like. Okay, so let's take this nanopore, and for the schematic that I'm showing, I will always show this kind of side view. So just remember that this is not a gap, but it's really a circular hole, for example, that has been just cut open to see from the side. So let's put it in the container here. Let's run this experiment again. So we attach the wires to the voltage source, and now the ions cannot just go any way they want anymore to the electrodes. They have to pass through the nanopore. The membrane is blocking any other pathway. So you can think of the nanopore as just a resistor. You can look at this just as a very simple circuit. You have a voltage source with current flowing. You can measure the current, and you have a resistor here. So Ohm's law is telling us that the current is inversely proportional to the resistance. So you see already now maybe the sensing principle. If I change something with the resistance here in the nanopore, I can measure this in the current. And that's exactly what we're doing. So now we have, in addition to the ions, I put here some purple blob, can be whatever. And if that moves into the nanopore during the experiment, then it affects the flow of the ions. It blocks the ions, it blocks the ionic current, and we will see a drop in the ionic current. So if we measure the ionic current as a function of time, if this blob, this purple blob thing, is moving to the nanopore, it increases the resistance, and so it reduces the electrical current, the ionic current. Okay, now, what if I don't just have a purple blob? What if I also have a green blob, a yellow blob, and a blue blob? And they all have different sizes. One is larger than the other always, so they will lead to different ionic currents. The larger the blob, the smaller the ionic current, because the resistance will be larger. So you can imagine then that you can calibrate your ionic current and basically say, okay, for this thing, the ionic current is this, and so on. So you know for different kind of entities, you have different ionic current levels. And then if these blobs are somehow connected by some kind of a strand and are pulled through, for example, if this is charged, if this thing that is attaching them has some electrical charge, then it will be pulled through. And you see this in the ionic current levels. So you get different levels, they're changing, they're going up and down. And by measuring the ionic current as a function of time, you can then reconstruct the sequence of what was going through the nanopore as a function of time. And that's exactly the principle behind nanopore-based DNA sequencing. So 4 different bases have different sizes, some are bigger than the others. So they block the ionic current to different degrees. And by measuring the ionic current as a function of time, you can then sequence DNA. In principle, I mean, this is the schematic. There's lots of technological problems with that, but we'll get into that later. Okay, one question maybe that comes to your mind is how to actually make these nanopores. I didn't really talk about this before. So there are different ways to make solid-state nanopores. One is that you use some kind of a focused beam, either electron beam or ion beam, and you have a thin membrane made of some insulating material like silicon, silicon nitride. And by focusing it, having a very small focus, you can actually knock out the atoms there, and you can form the pore. And it has some additional nice properties that I cannot talk too much about here now. But in principle, if you adjust the energy of the ions, everything okay? If you adjust the energy of the electrons or ions, you can actually even shrink the pore because you give energy to the atoms, and they can move into the pore once as they are heating the pore. So you can actually first make a bigger pore and then let it shrink and become smaller with this approach. Another approach that is very popular is controlled dielectric breakdown, where basically you always have some defects in the material. If you apply a high enough voltage, then these defects are starting to create more defects. Ions, the atoms in the material have electric charges, they're being accelerated. And so in the end, you create a big enough defect that the material breaks through, and then you notice this in the ionic current, and then through some circuit setup, you can automatically regulate that, then you turn off the voltage or reduce it so it does not create too big a nanopore. 2D materials are very interesting for nanopore sequencing, as I talked about before, because 2D materials have the kind of resolution to sequence individual bases very well, and there are different ways to do it. Either you have some kind of a 2D membrane, and then you transfer it on top of a silicon nitride membrane where some pore was created, or you use chemical vapor deposition, and you grow basically this 2D layer selectively on top of a pore. And there are different kinds of detection mechanisms. So I was just talking about before that you can use the ionic current. And this is, of course, something that is always possible. So if you have a nanopore and something is moving through the pore, you can always detect the ionic current here. But you can see that you can also have an electrical circuit, so you have an additional voltage source that is probing the entity that is going through the nanopore through an electrical current. And there are different ways to do it. One of them is, for example, that you have something going through between two electrodes, and then you have a tunneling current between the electrodes. And if something is moving between these electrodes, it bridges the gap, so it makes the tunneling current rise. And that is one way to sequence DNA. These electrodes can be embedded in a membrane, or you can have something where the current is passing by actually the pore. So you have a pore, and the current is flowing adjacent to the pore, very close to it. And then, just like in a field effector transistor setup, the conductance of this wire or place where the current is flowing nearby will be affected by what's going through, especially if it's charged, like DNA, and in that way you can sequence it. And so there was a nice experiment done in, I think, 2013 by the group of Alexander Radenovich, where they used the graphene nanoribbon, and it was attached to electrodes. So we have an electrical current running through here, and then you still have an ionic current running in the perpendicular direction. And when the DNA is moving through here, then you actually see that you get drops in the ionic current, just like I was describing before. So whenever the DNA molecule is going through very fast, so all of these negative spikes here, the drops in the ionic current, are due to DNA moving through. And simultaneously, you get positive spikes in the electronic current. And so the graphene nanoribbon temporarily has its conductance raised, and therefore this is an indication that you can detect also DNA through this electronic current here. Okay, so I want to talk now a little bit about what we are actually doing there in the division of materials theory to understand better these nanopore setups. So the advantage for us for doing materials theory, materials modeling, is that we have, of course, full control over it in the computer. So we're building models atom by atom, atomistic simulations, and then we can, through suitable approximations, through suitable models, explore them. So it's a little bit like sometimes people like to refer to it as a computational microscope. It's like a microscope where in the computer you basically observe a simulated system in as high resolution as you want, because we have full control over the system, like I said. So a typical protocol for these kind of explorations, for these kind of studies, looks like this. We start at the highest level with multi-dynamic simulations that can be force field simulations, but it could also be ab initio simulations, first principle simulations, where we have a distribution of atoms, we calculate the forces between the atoms, we update the positions from the accelerations and the velocities and the positions, and we do this every femtosecond time steps. So we have to use very short time steps to get the dynamics right. And then we repeat this for millions of time steps, or even tens of millions of time steps, to simulate the dynamics of the system on the range of picoseconds, thousands of picoseconds, even up to like tens of nanoseconds. If you have a powerful enough computer and the right computational architecture, you can even go to microseconds, maybe almost, but that's sort of the limit nowadays. Then we can take from these multi-dynamic simulations, we can take certain snapshots. Best is often to take uncorrelated snapshots. So in time, we take the configuration of all the atoms, and then we would like to find the electronic density and other properties of it. Often that system is too large to treat in its entirety with DFT. So we use an approach that is called QM/MM method, where we split up the system into a part that we just treat with force fields empirically with molecular mechanics methods. That's the majority of the system. And then a smaller part where ab initio is important, where quantum effects are important, we treat with density functional theory quantum mechanically. So in that way, we can obtain things like electronic density, total energies, forces, and also the Hamiltonian of the system in principle. And then we can feed the results from this calculation into something that is called the non-equilibrium Green's function method, and that can calculate for us the conductance of the system, or if we bias the system, if we apply a bias to the electrodes, we can also calculate finite currents for certain bias voltages. So, summarized again what I was just saying, we start with multi-dynamic simulations. In many of our studies, we have used GROMEX, in his simulation program. We use this to calculate the electronic structure using density functional theory for certain selected snapshots. Often we use siesta for this one, but not exclusively. And then we feed that one, then the results from DFT calculations, into the code for quantum transport calculation using this non-equilibrium Green's function method. And yeah, in the past, we have often used Smeagol for this one. Okay, now I want to discuss some examples that I've written about in the abstract that Rodrigo sent around maybe. So some selected papers I want to illustrate what this means in practice and what kind of results we get. So I was showing before on one of the previous slides that in 2010, people proposed to use graphene, graphene nanopores for DNA sequencing. So in 2011, we have published a study where we didn't look at graphene nanopores actually, but we looked at a graphene nanogap. So you have to imagine that you have a semi-infinite sheet of graphene here, and a semi-infinite sheet of graphene there, and they act as the left and the right electrode of our system. So we inject electrons from the left side here, and then through the gap, they tunnel to a molecule that's inside here. So this is one of the DNA nucleotides. And then from the orbitals there, they tunnel over to the right electrode. And we can calculate with density functional theory and this non-equilibrium Green's function method, we can calculate the probability for this to happen. So this is just like your very simple quantum mechanics scattering problem, just for a very complicated system, but basically this is what we can do here. So here we didn't do any multidynamic simulations. We just did static calculations. We put the 4 different nucleotides in this graphene nanogap and rotate them around only for different angles. But for frozen angles, then we calculated the probability for the electrons to make it from the left electrode to the right electrode. And then the results look something like this. So just to explain to you, the 4 different panels, they stand for the 4 different nucleotides being in the gap here. And the different colors of these curves here, they correspond to different rotation angles. And so then what we basically see here is that depending on the rotation angle, we get different coupling between the orbitals of the nucleotide and the graphene electrodes. And then for a finite bias, we can then, for example, integrate underneath these curves. For 1 volt bias, we would, for example, integrate from minus 0.5 eV below the Fermi energy to plus 0.5 eV over the Fermi energy, and integrate these curves, and then the area under this curve is proportional to the current that you would get from this. And then one can compare it, and basically the conclusion from the study was that yes, there's some overlap between these 4 different nucleotides, because the current can change over many orders of magnitude depending on how they're rotated, because it's tunneling current. But there's still, I mean, if you do statistics on it, you can probably distinguish between the 4 different bases. So this looks like a promising system. And another system that we studied a little while later was a bilayer graphene system, actually. So here we have not a graphene nanogap, but we actually have a source electrode, drain electrode. And then you can see here, there's a graphene sheet that is just connected to the source electrode, not to the drain electrode. And if we make a nanopore inside this graphene sheet, and then we have a second graphene sheet just below it, coupled to the drain electrode, that also has a pore aligned with it, and you can imagine that DNA can move through here, through these 2 pores, through these aligned pores, it's pulled through. And as this is being pulled through, it basically leads to a better coupling between the upper layer graphene and the lower layer graphene. So the distance between the 2 graphene layers is just right to couple only to 1 nucleobase at a time, because when graphene in the bilayer setup is stacked on top of each other, it's around 3.5 Ångström apart from each other, and that's also approximately the distance between adjacent bases in single-stranded DNA. And so for this study here, we did multidynamic simulations, and we didn't just calculate the conductance basically. So for different snapshots, all of these data points, these are basically snapshots for which the conductance was calculated. But in addition, we also determined the binding energy. We also determined how strongly do these bases interact with these bilayer graphene nanopore. And why do we do that? Because the translocation time, just go back 1 slide here, the duration of the signal is an indication of the translocation time. How long does it take the base to move through the nanopore? And so we can use both the information of the conductance change and the duration of the translocation time to identify the bases. And then we see here that adenine and guanine, they have conductance values and binding energy values that are quite distinct from each other. Cytosine is interesting in the sense that we have two very distinct regions, so that depends on the configuration of the cytosine molecule in the nanopore, actually. In some cases, we get very good coupling to the two layers, then we get high conductance. But in some cases, we get a configuration from the ND simulations that is actually not very good in terms of coupling the two layers to each other, so we get a very low conductance, almost zero basically. And then thymine is a special case. Thymine has a range of conductance values because it is a very small base, so it can rotate a lot in this nanopore, and so it overlaps also somewhat with the adenine and guanine regime. One more system that I'm very happy to talk about is a very nice collaboration both with Rodrigo and also Vandana, and this was what we call a basically a graphene nanorod, if you can see it here. So we have a graphene nanoribbon or nanorod, which is embedded in hexagonal boron nitride. So above and below, the graphene is limited by hexagonal boron nitride. Now, hexagonal boron nitride is not conducting. It's an insulator, has a gap. So if we have some left lead and a right lead, if we have electrodes that are injecting electrons again, then the electrons are forced to basically go only where the carbon atoms are, only where the graphene is. Now, in addition, we cut out here the graphene nanopore again, in just the graphene part, and we spare here an atomic wire of carbon atoms. And so the current actually is forced to go only here through this atomic wire of carbon atoms. And now you can imagine if something is moving through the nanopore, something is coming from out of the screen here and going through the screen, through the pore inside, then again we have this field effect transistor setup where the charges on this entity that's moving through can affect the conductance of this carbon wire here and then change the conductance thereby. So we tested this. So we tested this again. This was not ND simulations. This was just static calculations. But we did density functional theory, and then we did this non-equilibrium Green's function method on top. So for the 4 different bases, A, G, C, T, basically, for these kind of configurations, the side groups on the nucleobases, they affect the conductance of this carbon wire differently. In some cases, when we have a positive polarity, let's say for example for cytosine, then it enhances the conductance compared to the empty pore case. And in other cases, when we have something with negative polarity, like an oxygen atom close to the carbon wire here, then it reduces the conductance, just like in a field effect transistor. So it basically repels the electrons and it lowers the conductance thereby. So we see here, we can analyze this here for different gate voltages. So if we go away from the Fermi energy and shift the chemical potential, then we can say, okay, for certain kind of gate voltages, one can actually distinguish quite well here between the 4 different bases with this setup. Okay, so these were just some examples, basically, how one can use these kind of 2D materials and these kind of materials modeling to explore in the computer how to make this kind of nanotechnological devices for DNA sequencing. Now, actually, nanopore DNA sequencing is already a reality, but they're not using the systems that I've shown you here so far. There's a company, Oxford Nanopore Technologies in the UK, which in 2012, 2013, brought something on the market which can actually sequence DNA with nanopores. And they have since then, so this was 10 years ago, they have improved the technology even further. So now it's really a strong competitor compared to other DNA sequencing methods out there on the market. And what they are actually using is not this kind of solid state or 2-dimensional nanopores that I talked about. They are using biologically, naturally occurring nanopores, which are here schematically depicted as green entities, MSPA and alpha-hemolysin, which embed themselves automatically in some kind of a lipid bilayer membrane. So this is what I was saying about before. You have some insulating membrane, and then the nanopore is a tiny opening here in this nanopore complex here, basically, and where we have the shortest, where we have the smallest opening, that's the sensing region. That's where the ionic current is most affected by the DNA molecule moving through it, basically. And in practice, then one of the big problems in the beginning with this kind of approach was that the DNA would move through this nanopore very, very fast. And you could not detect the ionic current fast enough to really get some distinction between the base A, C, G, and T. But then some scientists and also this company had the idea to use an enzyme which is called phi-29 polymerase, DNA polymerase. And that enzyme, which occurs naturally in certain kind of bacteria, it has the property that if it grabs double-stranded DNA, it basically splits it apart into the two single strands. And the nice thing is that it propagates the DNA one base at a time. So it holds onto the DNA and it lets it only go one base at a time in a quite slow process. So it ratchets, one says it ratchets the DNA through there. And so you can imagine if you have this nanopore here, the polymerase is too big to fit through the nanopore opening here. And then the single-stranded DNA is sucked through the nanopore here, but it cannot be sucked through it very fast because it is held on by this polymerase here and it's tightly binding to the DNA and it doesn't fit through the nanopore. So by moving this double-stranded DNA one base at a time very slowly, it allows this DNA just to go through the nanopore very slowly, and that way you can read out the ionic current. And so, as I said, so this company have commercialized this and made this approach better and better to use, basically. And so one of the main advantages of this nanopore approach is that the device for using it is quite small. So it's not much bigger than the remote control that I'm using here to advance the slides, basically. And requires relatively little sample preparation. So the liquid containing the DNA that you want to sequence needs to be prepared, of course, somehow to get rid of some kind of contaminants and so on. So it would just clog the nanopore. But then you put it there on this microfluidic device here, and it is run then through an array of nanopores that actually in parallel sequence the genomes that are being run through the nanopore. And the cost is just around $1,000 US, so it's quite cheap compared to other methods. You can use it several times, but then after a while the nanopores become clogged and then you cannot use it anymore. Then you have to buy a new flow cell, basically here. And there have been some impressive uses for it in the field. So this is one of the advantages, that you don't have to need access to a big laboratory in principle. You can take a sample and determine the sequence right away. So in 2014 and 2016, respectively, there were this Ebola virus outbreak and the Zika virus outbreak, and it was used there actually to get some information, some almost in real-time information, how the virus is mutating and to analyze what kind of virus strands are currently prevalent in certain regions and so on. So in that way, it can help to save resources and save time. So it was some successful application of this Oxford nanopore technology device. Okay, so back one more time for the title, and I'm coming to the last part of the talk now. So DNA sequencing, one can say, is a solved problem. From a technological point of view, there is a product. Of course, there's still lots of fundamental research that one can do, how the DNA is interacting with the nanopores, and solid-state nanopores, and also these 2D materials that I talked about, could have some real advantage. But yeah, this is something for future discussion. But what is next, sort of, after DNA sequencing being solved? What is the next big topic that one can address with nanopores? And that is actually protein sequencing. And so there was an article in the beginning of this year where Nature every year somehow they picked like seven, the most promising technologies to look out for during the coming year. And in 2023, the first thing on the list was actually single molecule protein sequencing, and they mentioned nanopores in here also somewhere. So why is this so interesting? I mean, why are people interested in protein sequencing? The genome, the DNA sequence, it tells you in principle all the proteins that an organism can make, in principle. But it doesn't mean that it makes all of these proteins. And also it doesn't tell you in what magnitude does it produce these proteins. So life scientists are much more interested, if you take an individual cell, for example, at a certain point in time, how many proteins are there of the different kinds. Make some kind of a histogram, like of this protein, there are so and so many. Of this protein, there are so and so many. The relative ratios of that can tell you a lot about how the cell is functioning, and also if it is malfunctioning. In the case of diseases, maybe there's something wrong with the expression of certain proteins. So if you would have an approach where you can sort of detect and identify the proteins and count them very quickly, that would be tremendously helpful for life scientists and for healthcare. The problem is with proteins, you cannot do this so easily so far. To start with, you cannot amplify proteins. For DNA, there's a very simple method how to make many, many copies of the DNA molecule for sequencing. For proteins, you cannot do this. There's no mechanism to quickly copy it and make another proteins. You have to work with the material that is there. So nanopores again could play a role here, but it's very challenging. So the first big challenge is that for DNA, you only have to distinguish 4 different bases. But for amino acids, you have 21 different amino acids for proteins that are making up. So you have to distinguish between many different molecules here. On the one hand, it's easier because they come in very different sizes. Some have very big side groups, some are very small. But it's also very challenging because they have different polarities. Some are positively charged, some are negatively charged, some are not charged at all, or they have like plus and minus groups on the side. So even the translocation to the nanopore is not as simple as in the case of DNA, where you just have this nice negatively charged backbone that you could use to electrophoretically put it through the nanopore. For proteins, you don't have that, so you have to try some other ways. So in literature and in the field, there are basically 3 major strategies discussed how you could sequence proteins. The most simple, the most straightforward one, number 1 here, would be unfold the protein, make it just a linear chain of amino acids, and then somehow move that one through the nanopore. And you get an electrical current signal as a function of time, which then hopefully is indicative of what is the amino acid sequence. And by the way, if you have the amino acid sequence, then with advances in the field, for example, like AlphaFold or AlphaFold 2 from Google, you could probably construct the 3-dimensional structure of the protein. So if you have the amino acid sequence, then for many proteins, you would also automatically have the 3D structure, the folded structure, through approaches like AlphaFold. The second approach is through some kind of fluorescence. So I'm not going to talk much about this one, but basically you can tag just 3 different kinds of amino acids. The abbreviations for them are K, M, and C. And if you just tag those with different kinds of fluorophores, with different colors, different wavelengths, then just the sequence of the color signal that you get here is enough to uniquely identify the protein in like 95% of the cases. So if you go to the protein database and you search for what protein has a sequence K, M, C, and it doesn't even matter what amino acids are in between here, you can find out what protein it is basically. And then the last method is, yeah, you don't actually unfold the protein, you don't linearize it into an amino acid sequence, but you keep it folded and translocate that one through the nanopore. And you also still get some kind of current signal as a function of time. And that one you can use maybe not so much to identify the protein, you cannot maybe distinguish all the different proteins this way because they are too similar often, but you can learn some properties of the proteins, its shape, the volume, the charge, for example. And you can also study protein dynamics in this way. So just very briefly, I want to tell you one study that we did for protein identification, and then I want to show you some cutting-edge experimental study for protein sequencing, and then we'll conclude this talk. So what we have done here in this study is we looked at 2 different kind of mini proteins, so which are relatively short, which are relatively small, which have a similar kind of 3-dimensional shape, but they have different amino acid sequences, but they happen to fold in similar shapes. And we wanted to see if they can distinguish them based on the ionic current in the nanopore through this approach. So here's these 2 different mini proteins just rotated around from different point of views, and you can see, depending on which amino acids are on the surface, you have basically different polarities. The setup was as follows. So we had a silicon nitride nanopore, so not a 2-dimensional system here, but a silicon nitride core, and the mini protein inside here, counter ions present, water present, which is not shown, electrodes applying a 1-volt bias typically, and then measuring the ionic current measuring in the computer just means counting basically how many ions are passing through the pore as a function of time. And so if we do this, we get an ionic current that is very noisy. So we have to filter it to get something more stable. And then we can analyze that and do statistics, produce histograms for the two different kind of mini proteins. So blue is one and orange is the other mini protein. And you see here from this graph, for the bias voltage of 0.5 volt and 1.0 volt, because they have so similar shapes, it's very difficult to distinguish between them. Basically, they almost completely overlap the distribution of ionic currents. But as I said before, there's another way to detect something in a nanopore, and that is the duration of the signal. So in this study, then we try to understand or calculate how strongly do these two different mini proteins interact with the nanopore. So the nanopore is made of silicon nitride, and the proteins are made of amino acids. So we studied in the computer the interaction of the different amino acids that occur in the protein with a silicon nitride wall. So we use density functional theory for that. We basically just move the amino acid closer and further away from the silicon nitride slab and calculated the energy. And then fitting some Lennard-Jones potential to that one, we can determine the minimum and the asymptotic limit, and then we get the binding energy in principle. The binding energy alone is, however, not enough, so we also need to know what amino acids are at the surface, so we can basically quantify the exposure of the amino acids to the outside, and then how likely they would interact with the nanopore wall. So without going into all the details, we can combine this. We can combine the exposure to the outside of the protein together with the interaction energy and predict some range of interaction energy. And you see we have some large uncertainties here, basically, that can then be translated into a translocation time. So how much time would it take for this folded mini protein to translocate through the nanopore? And then we arrive at some results that are very different, order of magnitude different. So one mini protein takes of the order of 0.4 microseconds and up to, well, in a range from 0.4 to 3 microseconds, while the other one would translocate much faster because it's not interacting so strongly with the pore. And so one can distinguish them based on the translocation time. And then the last thing, as I said, was this cutting-edge experimental study from Kees Dekker's group in the Netherlands, where they have experimentally studied the linearized protein, so a chain of amino acids, and get some kind of electrical current signal there. So how did they do this? So this is from 2021. The approach is they use DNA to actually pull the peptide through there. So they have a peptide, which is these amino acids, connected to a DNA strand. Just to make it simple, let's just say like that. On the DNA strand, they have something very similar to this polymerase that I talked about before. It's a helicase. Helicase is normally used to unwrap double-stranded DNA in the chromosome, but here, basically, it's just attached to the DNA here, and then when this string here with the polymer and the DNA is sucked into the nanopore, then again, just like before for the polymerase, the helicase cannot move through, but once it sits there, it starts to ratchet the DNA up. It moves the DNA in this direction, so it pulls everything out of the nanopore again, and eventually then you get to the point where it pulls out the peptide. So then you're reading one amino acid at a time, the ionic current through the nanopore here. And you get different levels then in the ionic current that can be indicative of what amino acid you have there. And they're actually shown in this paper that yes, you can in this way distinguish between. So here's a more simplified cartoon just to explain this method again. So you have this helicase bound to the DNA, and it's here it's shown like a wheel. So it's basically it ratchets up the DNA in this direction, moving the peptide out of the nanopore, and you read these different ionic currents for the different amino acids here. Once the helicase gets to the polymer here, to this peptide, it falls off because it's only designed to work with DNA. So it doesn't know what to do with amino acids, it just falls off. But then there's another helicase in solution. There's many of these helicase enzymes in solution, and they attach to DNA again, and then the process repeats. So it's always then pulled through again until the helicase sits on top of the nanopore, it pulls out again, and then the process repeats. So you can read the polymer, you can read the peptide chain as many times as you want almost, and in that way you can reduce the error rate. Basically, you have almost 100% certainty after a certain number of reads here to have identified the amino acid correctly. Okay, I think it's time to conclude because I don't know exactly how long I talked, but maybe too long. So, quick summary for the two parts here. So, the nanopore-based DNA sequencing, as I said to you, it's a cheap method. One advantage compared to other methods is also you can read very long stretches of DNA. I think the record is now 4 million bases in a row, basically. So that makes it easier then to assemble the entire genome because the puzzle pieces are larger, basically, and therefore fewer. No amplification is required. It works in principle just with the genetic material that is there in the sample. Modified bases, for example, epigenetic modifications could be directly detected. For the protein studies with nanopores, as I've shown you, so it allows to detect and characterize proteins again at the single molecule level, which is something that scientists are very much interested in, and you can get information on a whole range of protein properties. I'm not going to read this whole list here, but basically you can really do very interesting science on individual proteins. And what's the future holding then? So 20 years ago, I mean, this kind of DNA sequencing device that we have now accessible would have seemed like science fiction. So I told you that, I mean, it took hundreds of millions of dollars and years and the effort of many labs and groups working together. And now here we are today, and we have this MXene device, for example, that can do it in a couple of hours, less than a day, certainly, with just one person operating it, and for a cost of $1,000. So we should not underestimate how fast technology develops. People who are working in this field are not saying that whether it will be possible to sequence proteins with nanopores, but it's really just like when will it be possible? How soon will it be possible? So very likely in the very near future, we will see nanopore technology for studying the project. Okay, and for this 2D materials part that I talked about, our own studies, 2D materials have certain advantages for studying biomolecules, and in particular with this kind of hybrid materials. So if you have graphene and boron nitride, there's so many different combinations that you can play with, and there's so many other kind of 2D hybrid materials that one can envision for nanopore sequencing. So has a very high chance to create something that is very advantageous compared to other solid state or biological nanopores. Okay, finally, the most important thing, acknowledgements. So this would not have been possible without lots of collaborations and lots of people working with me and doing the calculations. I put Rodrigo there first because, as he mentioned, I know him since 2005, and we have worked together for many, many years, and he's one of the most hardworking and productive scientists I know. Alejandro also, I met only short time after I met Rodrigo, I met Alejandro, and he's also been working with me. We've been working together for a long time. And there's a whole list of Brazilians here. I'm not going to read all the names, but Vandala was also instrumental in some of the studies that we have done. Jarani here, she was a PhD student in our group. She's now back in Thailand, also very much there from the beginning. And then more collaborators here, and I'm not going to read all the names. Funding, and then as mentioned, Capes for bringing me here to Volta Redonda, and finally, obrigado for the attention. Thank you.
Thank you all for the very nice talk. Questions?
I have one. I wish thank you for your talk. It was very nice one. The slides were certainly beautiful. And I have a question about the geometry of the nanopore.
Yeah.
Let's talk about the graphene and the UK companies. The UK companies are biological. One of the graphenes is official. I wish to know how does the difference of the geometry of the nanopore influence on the DNA sequence detection?
Right. So I think the most important feature, as sort of explained here, is that the sensing region needs to be as small as possible, and it needs to be narrow. So if you use biological pores, they also have very similar dimensions like the solid state pores. So the diameter of this opening is also like of the order of 2 nanometers, let's say 2 to 3 nanometers at the most. And then the sensing region, basically the sensing region tells you how many nucleobases in a row you are simultaneously measuring. So I didn't tell the whole story here. Actually, for this Oxford Nanopore Technologies, they don't have single base resolution. They always get the signal from 3 bases at a time, but then through some clever algorithms, and because you always know that one base is moving out and the next base is moving in, you can actually then decode the base sequence this way anyway. So one of the advantages for 2D materials is that the size of the pore opening is the same as in the biological pores, but the thickness is in principle 0. In principle, it's like as thin as it gets, just 1 atomic layer thick, or even the bilayer graphene is very, very thin. So one of the advantages of two-dimensional or 2D materials for nanopore sequencing would be that you would have this single base resolution. There are many problems to address that make it not so attractive, and that's why it has not been realized yet. But if it can be realized, then the single base resolution is one of the most important positive features of this technology. Did I answer the question?
Yeah.
Okay.
For the Nano-Institute information, you showed the system which has 2 layers of graphene and okay. So how do you set the distance such that the graphene layers don't interact if they're not supposed to interact with each other?
Yeah. So it's actually it's like this, that they are stacked on top of each other. So it's like pi stacking, AB stacking, for example. And so in principle, you do always get some small tunneling current from one graphene layer to the other. That would be unavoidable. But if a DNA strand is moving through, it's a little bit hard to see here, but basically the edge of the upper layer graphene nanopore couples electrically to the base there, and then through the molecular orbitals, it is conducted to the lower part here, and then again it is connecting to the graphene nanopore here. And that gives rise to a much larger current, because always the tunneling distance is much less than the 3.5 angstrom here. Maybe the only thing to be concerned about is that if you would have a very large overlap, so if this is a big graphene nanoribbon and the pore is relatively small, then maybe the background current, the base current is very large and you have a lot of noise in it. Then maybe it would be hard to detect it with this. So the approach would have to be that you make the overlap as small as possible. You could think, for example, that you have 2 graphene nanoribbons, and instead of overlapping it like this, you could maybe cross them, for example, and make the nanopore where they're crossing. Then the overlap would be less. So that would be one approach. Yeah, so it's a very good question.
Okay.
Excuse me, Professor, thank you for the talk. It's really great. In the first graphs, you have showed some spikes, but I didn't get it, the physical interpretation about it, the relation with the model you have proposed.
Okay.
Would you mind to explain it again?
Was this slide? Yeah. Okay, I see. So in this setup here, this is real experimental data. So they have used this ionic current. So you have basically the side view, just like I've shown before. You have always positive ions going this way, negative ions going that way. And this noise here, what you see here, that is basically just when the pore is open. When this graphene nanopore here is open and there's nothing inside, no DNA inside, just water and ions, then you get this very noisy ionic current, basically. And when the DNA is moving through, when it's being pulled through, then for a very short time, just like a blip, and then it's already through, the ionic current gets temporarily reduced a little bit. It blocks you a little bit. And at the same time, they measure in this direction. They have another set of electrodes. It's gold chromium or something, deposited on graphene. And so we always have a current running through this graphene nanoribbon from one side to the other. It's also quite noisy, as you can see here. But again, every once in a while, suddenly for a very short time, the current becomes larger than this baseline current. And it's always correlated to this ionic current event. So it means it's very, convincing that it's due to the DNA either moving through the pore, or what can also happen, because this is a time scale of seconds, the DNA can move around in motion in the liquid. It comes close to the nanopore, temporarily blocks it, and enhances the electronic current, and then moves away again. So it could also be that it's not really moving through the nanopore, but it's just close to the nanopore and affects the conductance, both from the ionic current and the electronic current.
So that noise is sharp noise, and the other spikes are the meaning of it.
Yes, exactly. Yeah. Yeah.
It's very nice because they have two different mechanisms.
Yeah.
And they have the correlation between them.
Yeah.
And there's different levels.
It was what I wanted to ask. If they are, you are ensuring that the spike over there is related to the DNA, not because you have like a second information from the electronic charge, right?
Right. Yes. Yes.
Like there's another spikes, small spikes there that are not shown, but
Yes.
that they are not linked with any spike from the electronic charge, so you don't have the.
Exactly. Yeah. Right. Yeah.
It's nice.
Exactly. So it's. I mean, in most of the events there's some correlation, but not in all of them. And of course, I mean, there's a lot of things you have to do to rule out that it's not something in the circuitry that is like influencing each other. Because there are two circuits that are very close by, and this is a very small setup, of course. But yeah, I think they did very careful study here, so it's probably really something moving into the nanopore or close to the nanopore.
Any other question? Talk. I'm curious because to determine 4 bases for DNA is okay. You have 4 steps of
Yes.
programming, but for proteins,
Yes.
there's only one. Do you have a resolution?
It seems
Maybe
like
5.
I mean, it seems possible. So this last study that I was talking about here, this is really experimental data, and there's also at the lower bottom only here, there's some predicted from MD simulations actually. And so I don't know whether they can really distinguish all 21 amino acids yet in this way, but at least with this approach, which is very similar to the approach that Oxford Nanopore Technologies is using by moving it like one base at a time, in this case, in 1 amino acid at a time. If it's long enough in the pore, then you can really you sample of the ionic curve long enough that you can distinguish apparently between these 21 levels. But I agree with you. I mean, I also think it's very hard to believe that you can get so many different ionic current levels and make them out under the noise, basically. So, I mean, one of the things we discussed just yesterday, actually, in Sao Paulo, is that maybe you can also use something to couple to the amino acids to sort of make the fluctuations less or make the interaction with the nanopore more specific so that you can distinguish better between these 21 amino acids. But yeah, it's very challenging, but everybody who's working in the field, including me, is very optimistic that we can overcome these challenges somehow and so.
There's another schemes or another proposal different from nanopores for protein.
I mean, what people have been using so far is mostly this. What is it called? Mostly it's being done by this kind of mass spectrometry so far. That's what most proteomic analysis so far is using, basically. And there are some other approaches also. Actually, in this article, they're also discussing other ways with fluorescence actually to tag the amino acids somehow to identify the sequence order. So there are other approaches also, I should say, not just nano.
Again, but right there where you was talking about this, because sometimes you have the charge of electricity to measure, but sometimes you need to also the speed, the time of the reaction, right? And when you're using this for protein, you have the thing that slows down the flow of the protein there in the.
Yes, that's right.
So maybe it can mess a bit up with the precision you can classify them because you don't have the time parameter.
Good question, exactly. So in that case, then you don't distinguish based on the duration of the event, but just purely on the current level. So it would be nice to have some way to slow down the translocation of both DNA and proteins without having this kind of external mechanism that just always moves them through with the same speed. So I think it's exactly what you're asking. It's a very good idea. If you could come up with some other methods to slow down the translocation so that you can read the signal in a stable way, but it also is distinguishable then for the different amino acids, for example. That would be very nice. And yeah, I think this is something that we are working on, discussing with the experimentalists and trying to come up from the theoretical side of new devices that could potentially be.
Sometimes it just maybe it's just a clever algorithm to do the reverse engineering to find it.
That's true. I mean, there's a lot of also for this company. I mean, I think they're also using machine learning now to identify the signals and then basically say, okay, this is this entity and so on. So if you have enough data and together with machine learning, yeah, this could open new approaches also for the identification for sure.
Because you indeed, it's like you need to use machine learning to solve a problem that can train another machine learning to solve another problem, like portraying simulation.
Yes, that's right. Okay, thanks for that.